Cell division via mitosis. The first step to this process is prophase. At this point, the DNA has already been repeated when the cell enters this phase. Chromatin fibers condense, nucleoli disappear, and chromosomes appear as two sister chromatids are joined at the centromeres. We then move to prometaphase. This is when the nuclear membrane breaks apart. Kinetochores form at the centromere of each chromosome, and some microtubules attach to these kinetochores and the chromosomes. After prometaphase, we move into metaphase, and this is when the chromosomes line up in the middle of the cell at the metaphase plate. After metaphase, anaphase. This phase is the shortest phase, clearing all cohesive proteins and making each independent chromatid become a single chromosome. The new daughter cells begin moving towards the end of the cell. After anaphase is telophase. The two daughter nuclei form at the cell and the nuclei reappear. Cytokinesis. A cleavage furrow pinches the cell in two, making two completely different cells. Cell division via meiosis. The first step of meiosis is prophase number 1. At this point, each chromosome coils and becomes shorter and thicker. This is the longest phase of meiosis, and crossing over occurs, which gives it its own genetic combination. After prophase 1, we move into metaphase 1. Pairs of homologous chromosomes are arranged at the metaphase plate. After this step takes place, we move to anaphase 1. In this step, proteins responsible for joining the sister chromatids break down and allows the separation of the chromatids. However, sister chromatid cohesion remain at the centromeres, causing chromatids to move as one unit. We then move to telophase and cytokinesis. At this point, the homologous pairs complete their migration to the two poles. We now have two haploid set of chromosomes at each pole. Each chromosome has two chromatids, and then at cytokinesis, pinching of the cell results in two cells. We then move to prophase 2. At this point, chromosomes made of two chromatids are moved by microtubules to the metaphase 2 plate. In metaphase 2, chromosomes are positioned on the metaphase plate. Two sister chromosomes are not genetically identical, and the kinetochores of sister chromatids are attached to microtubules extending from the opposite poles. After metaphase 2, we move to anaphase 2. At this point, the breakdown of proteins holding the sister chromatids together at the chromosomes allow the chromatids to separate. The chromatids move towards opposite poles as individual chromosomes. After anaphase 2, we move to telophase and cytokinesis 2. At this point, the nuclei form, chromosomes begin condensing, and cytokinesis occurs. Mitotic diffusion for one parent cell produces 4 daughter cells, each with a haploid set of chromosomes.